is our chief guest of this session, sir.
Okay, ma'am. Thank you.
Okay,
Thank
sir.
you, Dr. Maithili.
Thank you, sir.
A very pleasant morning to one and all present here. This is Professor G. Mohan from the Department of Mathematics, KSRCT. It's my privilege and honor to introduce today's session 2 Chief Guest Dr. V.Shanmugaiah, Assistant Professor, Department of Microbial Technology, School of Biological Sciences, Madurai, Kamaraj University, Madurai. In the year 1999, he completed his bachelor's degree at Vivekananda College, Thiruvegadagam, Madurai. In the year 2001, he completed his master's degree at St. John's College, Palayamkottai, Tirunelveli. In the year 2002, he completed his MPhil degree at VH College. In the year 2007, he completed his PhD at the Center for Advanced Studies in Botany, University of Madras. He has received advanced studies awarded by Vivekananda College from 2004 to 2005 under the Earn While Learn scheme of the UGC program. He has worked as technical assistant at the Center for Advanced Studies in Botany, University of Madras. From January 2008 to 2009, December 2009, he was working as postdoctoral fellow at the Department of Biology, Disease and Biotechnology, Agroforestry, University, Italy. In the year 2011, he received Young Scientist Travel Grant awarded by Federation of European Microbiologists. He was awarded as Raman Postdoctoral Fellow by UGC for the year 2014-2015, and he was working at the Department of Biology, Rutgers University, New Jersey from November 2014 to November 2015. He is very much interested to do research in the following areas, and they are molecular plant, microbe interaction, investigation of antibiotic genes from
Oh, sir, your mic is still mute. Oh, sir. Your microphone is muted.
He is very much interested in to do research in the following areas. They are molecular plant microbe interaction, investigation of antibiotic genes from microbes, plant defense mechanisms, nanoparticles application as biopesticides, antimicrobial substance extracted from bacteria, actinomycetes, fungi and medicinal plants and their activity against plant and human pathogens. He is a life member in different scientific bodies, and they are life member in Biotech Research Society of India, life member Academy of Biological Sciences, life member Association of Biotechnologists of India, life member in Association of Microbiologists of India, life member in Mycological Association of India, life member in Phytopathological Association of United Kingdom. He has visited 4 countries, Italy, Germany, Switzerland, and USA. He has presented his research findings in 16 international conferences and in 13 national conferences. Totally, he has published 43 research articles, 40 in international journals, and 3 in national journals. And finally, I would like to thank the management, principal, the head of the Department of Biotechnology of KSRCT, my dear colleagues, and the organizers of NSF for giving me this opportunity. Thank you once again. On behalf of everyone, I invite today's chief guest, Dr. V.Shanmugaiah, to the session. And now, I hand over the session to Dr. V.Shanmugaiah. Thank you, everyone.
Thank you, sir, for your nice introduction about me.
So, thank you so much.
Thank you, sir.
Sir, you can start slide sharing, sir.
Yes. Now, can I start?
Yes, sir.
Okay. So, I would say very good afternoon to all. First of all, I would like to thank the organizing committee for organizing this wonderful event for 25 days faculty development program. Respected organizing committee members, my dear participants, students and friends, very good afternoon to all. I am very happy to present my topic entitled on methods in plant disease control. You know, the plants, they are affected by, there are 2 important factors. One is biotic and abiotic factors. The biotic factors means, see, for example, so many microorganisms, for example, bacteria, fungi, viruses, and nematodes. They are effectively.
Sir?
Hello? Hello?
Screen is not shared right now, sir.
Why?
Screen is not coming. Screen is not coming.
Now?
No. PowerPoint display is not coming.
Sir, you just go to the Teams and then you put an upward arrow mark. There will be an upward arrow mark, sir. From that, you have to open the share window and then you have to show the desktop.
Share, okay. Share.
Share, you'll have desktop and then PowerPoint, which is open, that one. Okay, I think it's going to get shared.
Now it's okay.
I think it takes few seconds to switch over, sir.
Not coming.
No, it's not shared right now. Sir, as we did in the morning, first you have to open your PowerPoint in the desktop.
Yeah.
Yeah. And then you have to enter into the Teams and then go for sharing.
Okay, hello.
I think it will get shared, sir. If it is shared, I will inform you, sir.
Okay.
So far, it is not shared, sir. Sir, and the share screen button, ungalku PPT keyla desktop theriyudunga, sir? Desktop என்ற ஒரு option.
1 second. Share, okay.
Share கீழ வந்து ஒரு pop-up window open
ஆகும். We have to on camera, no? Turn camera. Camera, okay?
Yeah, that is okay, sir. That is okay. Yeah, camera is okay. அதுக்கு next வந்து microphone இருக்கும், share இருக்கும். அந்த share option click பண்ணீங்கன்னா
okay.
கீழ வந்து உங்களுக்கு desktop, windows, screen 1 அது மாதிரிலாம் காமிக்கும்.
Desktop நான் click பண்ணிட்டேன். Desktop.
Okay.
Hello.
Yes, sir. இப்ப desktop தெரியுது. இப்ப நீங்க அந்த PowerPoint முன்னாடி வச்சிருந்தீங்கல்ல?
ஆமா.
அது தெரியணும் சார். PowerPoint ஓபன்ல வச்சிருக்கீங்களா desktopல?
ஓபன்ல.
அது கிளிக் பண்ணுங்க சார். அது கிளிக் பண்ணுங்க. PowerPoint கிளிக் பண்ணுங்க. Yes sir. Yes sir. We can see right now.
Okay.
F5 போட்டீங்கன்னா full screen ஆயிடும் சார்.
Oh, okay.
I think it's taking time. No problems. No problem, sir. நீங்க அப்படியே கூட கண்டினியூ பண்ணலாம். Next slide மட்டும். Yes, sir. Yes.
Okay.
We can see full slide. Yes, sir.
So,
okay, sir. You can start the session right now.
Yeah. So, plants, they are affected by two important factors, biotic and the abiotic factors. The biotic factors means there are so many microorganisms affected by plants, for example, bacteria, fungi, virus, and nematodes. So in India is an agrarian country, overall 65% of our country depends upon the agriculture, and also 45% of our Indian economy rely on agriculture based. So biotic factor, to control the biotic factor is a major limiting conditions in our country. Apart from that, abiotic factors, so many physical and chemical factors also play important role for the engagement and development of the disease. So we need to develop the alternative methods to control plant disease. So based on that, I have chosen the following this topic: methods in plant disease control. So in this talk, we are going to discuss methods in plant disease control. What is biological control? What are the benefits to it use? Then mechanism of biological control, requirement of successful biocontrol, working example of biocontrol. See, there are, in general, in those days, people are using, there are 4 methods for the control of plant disease: cultural method, physical method, chemical method, and plant quarantine. See, cultural methods means for the cultivation of plant, the selection of geographical area is most important, because in the geographical condition, the temperature and humidity are very important to require for the development of the plants. See, for example, temperature and humidity for a particular crop, for example, fungi and bacteria. Some of the fungi, they could grow in a very humidity condition. Some of the bacteria, they could grow in a very temperate condition. So geographical location is very important. Most of the soil-borne disease are controlled by proper selection of the field, because some of the microorganisms, they very well in a particular field, because it is we could reduce the based on the selection of proper selection of the field. See, for example, you may take Calotrophium falcatum. It is a red rot of sugarcane pathogen. This pathogen, they could survive in a humidity field condition. Or also you may take in Rhizotonia solanae, this fungi cause sheet blight of rice. These fungi also, they may grow in a very humidity condition. So selection of field is also most important. Then choice of time, sowing of the seed. See, for example, sowing of the seed also, it is most important for the control of plant disease. And a susceptible stage of plant growth, and the pathogen should not match at the same time, because some of the microorganisms, some of the pathogenic microorganisms, they may grow in a particular environmental condition. We should not match during the sowing condition, sowing time. And moreover, whenever we start to cultivate the plant, we have to eradicate the pathogen contained seeds. We have to eliminate the pathogen first. So then only we need to start to showing the seed. See, for example, you may see some of the plants, for example, there are some of the disease, for example, powdery mildew and rust disease, because easily growing some of the plants, for example, easily early maturing variety, for example, pea is capable of escaping powdery mildew and rust disease. Then crop rotation also play important for the control of plant disease, and selection of the seed also is the most important, because we must avoid the seed-borne disease. Unhealthy and disease-free seed are essential, and we have to destruct the diseased seed. And even, for example, suppose a plant is affected by a disease, you must remove the organ of the plant, or we must remove whole plant also from the field, through which we could control the disease. Then selection of the seed, avoid the seed-borne disease, and the disease-free seeds are essential. Then modification of cultural practice. See, while cultivating the plant, showing the seeds and transplantation, the distance between the plants is also most important. Time of frequency, the irrigation frequency, transplantation method, and mixed cropping, and utilization of the amount of fertilizer and compost. These are all play an important role to reduce the disease caused by microorganism. This is the one method. The next method is physical method. See, disease caused for the control, the physical method is developed in the year of 1887. So disease caused by the control of smut disease, wheat, barley, and goats. Those days, people are use solar energy and hot air to minimize those diseases. Then chemical method. Chemical method also people those days used to control most of the diseases. Seed treatment with the fungicide before transplanting. Soil treatment. It is used for controlling such as joil pond disease which attack in seeds or seedlings. For example, such as chemicals, you may take the formaldehyde, capton, thyrum, organomercurials, PCNP, ethylene, dibromide, weapon, etc. For externally used seed-borne disease chemicals such as formalin, copper carbonate, capton, organomercurials are used for seed treatment. For internal, then systemic organic compounds. See, for example, those days before developing the systemic fungicide, people are using so many chemicals for controlling so many airborne diseases, foliar application of chemicals also. The common copper fungicide, Veranex, Beryllium, Philitox, so the Phytalon, these are all used comparatively better than that of products mixture. Seed dressing with organomiracles and systemic fungicides. But even though those fungicides cause severe threat to the environment and public health, then only nowadays people start to use only the organic biofertilizer and microorganism for the control of various diseases. Then plant quarantine. So plant quarantine, one kind of method to minimize the pathogenic microorganisms spread from one place to other place. I mean, from one continent to another continent, because the plant quarantine, through the plant quarantine, we could minimize the plant pathogen from escape from one place to another place. I mean, the movement, restrictions on the movement of agricultural commodities for the purpose of exclusion and prevention or delay the establishment of plant-based disease in areas where they are not known to occur. So with these, there are 4 methods are finished for the plant disease control. But mainly, nowadays we are focused on, most of the scientists nowadays to focus on biological control. The biological control word, the first coined by the Harry Smith in relation to the biological control of bacteria, fungi, and insects. So, what is mean by biological control? The suppression of pathogenic bacteria, fungi, and insects population by native microorganisms or introduce other microbes isolated from different habitat. Why we are using biological control? Because biological control is a very easiest way. Whereas suppose you may use the chemical pesticide, they are expensive, labor intensive, cost specific. Whereas in biological control agents are cost effective, easy to apply, and broad spectrum. They could control various diseases. One single microorganism, they have a broad spectral activity. So in our lab, we are doing isolation of fluorescent pseudomonas from various microflora, various rhizosphere origin, rhizosphere habitat, and then we detect the antibiotic gene and their regulation from our isolates, and through associated free-living bacteria like Pseudomonas, Bacillus, and actinomycetes. Then we are also studying the interaction between microbial community and the interaction between plants, and they control the plant disease control. Then why we are mainly focused on rhizosphere? Because almost 50% of our plant biomass, they are present inside the soil. Because inside the soil, the fantastic biochemical nature is going on, function is going on. For example, you may take the rhizosphere. So rhizosphere is those soils are present in and around the roots, they are called rhizosphere. Because the rhizosphere, there are millions of microorganisms are there. Because the root roots, they are produce some antibiotics, some sugar substance, they are called root exudates. Through using that root exudates, the microorganisms, they had enhanced the production of so many nutrients through the transportation, through the water transportation, the microorganism, they expose their product through the root system, through water transportation. So rhizosphere, generally the rhizosphere is a dynamic environment. The plants, they extract 70% of fixed carbon through their roots. So, 50% of biomass, they are present inside the soil, because they could, most of the plants, they are through the root system only, it is enhanced their plant growth, because plant growth hormones is developed inside the soil because of the root exudates. So what is determined the disease? So plant, for example, you may take the plants, various species of plants, various genotypes, various stages of the growth, then various stresses are involved. Then the pathogen. So pathogens, various genotype, then hypersensitive response, avirulent response, various races of plants, pathogens, and then the dispersal capacity and colonization site. Then environment also play important role. Humidity, water, plant factors, and the other microbes. See, for example, you may take the firefly disease in apple. This apple plant, we could cultivate only the highly humidity areas and hill areas. So those areas, the microorganisms, they could, because fungi, they could easily grow in a humidity area. So then only the apple plant is affected by fire blight disease. Because most of the pathogens, they create so many diseases through which annually 1.2 to 1.3 trillion crop protection worldwide. So, but losses is 500 billion. Then additional losses without current control methods, 330 billion. Then coming to the rice, rice is a most important crop all over the world, especially tropically in countries like India and China. These rice plants, they are affected by so many bacterial and fungal pathogens. So major pathogens, for example, Pyracloria rizae, Rhizotonia solanae, and so many fungal pathogens. And coming to the bacterial pathogen also play important role, cause various diseases. For example, Xanthomonas oryzae. First, we talk about the leaf blast disease. Leaf blast disease caused by Pyracloria rizae. You could see here the spindle-shaped spots appear on the leaves. The shape is caused by the Pyracloria rizae. Once it is developed, this kind of eye spindle shape appear on the leaves. So it is disrupt the photosynthesis and translocation of food material from one part to another parts of the plant. Once it is, because the xylem and vessels, they are going to disrupt the exposure, nutrient transportation. Once it is developed, this kind of spindle shape means the tissues is going to decay because of the development of this kind of the symptom. You could see the brown margin and leaves and enlarge, and finally they kill the leaves. Then node blast. It is also caused by the same pathogen, that is Pyracloria rizae. It is also infected the node region of plants, node region of rice plants. Then irregular lesion is developed, and it is also infected nodal parts. Once it is infected, the nodal parts going to leads to death. Then neck blast. So next blast, it is also same pathogen. It's grayish brown lesion of neck. Discoloration. You could see the discoloration of grain after infection, and quality of grain or form. So why I'm showing this kind of the disease symptom? Because a single plant affected by various pathogen. You could see any rice plant itself is caused by various diseases by various microorganisms, various fungi. For example, in sheath rot caused by Sarocladium oryzae. The Sarocladium oryzae, it has developed the irregular spots on leaf sheath, chocolate brown in color. It occurs during the maturity stage. Once it is maturity stage, is developed this disease sheath rot, and then the yield is going to loss, then discoloration and dried of the grains. Then false smut. False smut caused by another kind of Stilla cyanoidea virens. It is occurrence of smut after flowering. The appearance of yellowish and short ball or grain. Few grains are infected and the rest of the normal. It is found mainly in Haryana and Punjab area. Coming to the sheath blight of rice. Sheath blight of rice is a very economically important disease caused by Rhizoctonia solanae. Because, it is a Rhizoctonia solanae, it is a very important disease. It comes under the Detromycid group of fungi. These fungi, it is withstand in our soil in a decades. Because once it is introduced in our agriculture field, this Rhizoctonia solanae, it could produce a fruiting body called sclerotium. Because the Rhizoctonia solanae, they could develop, there are 3 different kinds of hybae. For example, lobate hybae, irregular hybae, and molanoid cells. The molanoid cells, they produce the fruiting body called sclerotium. That sclerotium, they withstand in our soil, in our agriculture field in a decades. We cannot easily eradicate this pathogen. So it is a highly, very important, economically important disease. Because due to this Rhizoctonia solanae, yield loss up to 69%. Then Tekal disease of wheat, it is a very, very economically important disease. It is especially in United States of America. This disease caused by Quanomyces graminis triticus. It is also a very important and essential disease. See how the Quanomyces graminis triticus develop. This Quanomyces triticus, it is in fact the root portion of the plant. Because once it is inverts inside the root plants, it is destroyed the vascular disease of root. Once it is destroyed the vascular disease, for example, xylem and phloem, once it is affect those kind of the vascular bundle, that nutrient exchange from root to shoot, it is going to arrest. So through which the plant is going to wilt, then the Tekal disease is established. So this is how the Tekal disease are present conductive soil and suppressive soil. So mainly it is present in the mostly suppressive soil. Yeah, you could see here, this is Rhizoctonia solanae morphology in a petriplates. You could see this here and there some arrow-like things are there. These are all called sclerotium. It is nothing but fruiting body of sclerotium. It's sclerotium because it's initially the Rhizoctonia solanae, it is it is a white color, then it is turned brown color. Then Macrophomina phaseolina. It is also another very important soil-borne pathogen, Macrophomina phaseolina. Macrophomina phaseolina, it has caused various root rot diseases and so many wilt diseases also. Yeah. Then we are isolate so many microorganisms. Especially, first we are going to discuss the actinomycetes. We isolate the actinobacteria from rice rhizosphere. So you could see these are all some of the isolates of our actinobacteria from rice rhizosphere. Then we check out towards all the isolates of our actinobacteria towards our target pathogen, that is Rhizoctonia solanae, by dual culture assay method on potato dextrose agar medium. Because those days the people are using physical, chemical and cultural methods. Those methods is not successful. But we have to develop a new microorganism. For that, we have to develop you find out a potential microorganism. So for that, we have to isolate so many number of microorganisms. From that, we find out a potential microorganism based on their zone of incubation, based on their control potential against our target pathogen, that is Rhizoctonia solanae. Yeah. So totally we isolate, there are some about 100 isolates of actinomycetes. Then among 100 isolates, we find out of only one microorganism, that is VS-MGT1014. This microorganism, it is a significantly controlled the Rhizoctonia solanae. The Rhizoctonia solanae, we could grow already. There are 14 difference of medium, because ISB 1 to ISB 14. It could grow very well in the international streptomyces project medium. A specific medium for growth of the streptomyces, that is from ISB 1 to ISB 14, but we use, there are ISB 1 to ISB 7. But the color range, the substrate color and the nature, morphology of the color also going to change from medium to medium. See, you could see in a substrate mycelium, when you are grow on the ISB 1, it is gray color. Whereas you grow in a Pinnett's medium, it is yellowish brown. So that's a way the morphology also going to change different medium to medium. Then we have checked out our selected microorganism VS-MGT1014 against various fungal pathogen, that is Rhizoctonia solanae, Macrophomina phaseolina, Caesarium oxysporum, and Caesarium budum, and Alternaria alternata. So using dual culture A as well as well diffusion assay we have done. You could see here the zone of incubation, that is the Rhizoctonia solanae, that is 30 millimeter to 18, from 16 millimeter to or to 30 millimeter. Whereas you could see the cell-free culture filtrate of our Streptomyces VS-MGT1014 remarkably control various pathogens, starting from Rhizoctonia solanae to Alternaria alternata, with the different level of zone of incubation, using various concentration of cell-free culture filtrate compared to control. You could see the, in biological control, there are three main mechanism. One is antibiosis, parasitism, another one is competition. Then how our microorganism to control this particular pathogen, we need to find out. In our laboratory itself, we could find out, just here take a pinch of mycelium from the dual plates from the zone of incubation, and then you could check on the just a glass slides, and then strain with a cotton blue, and then you could see under the light microscope, you could see here the destruction of the mycelia of Rhizoctonia solanae, and then some disintegration of mycelium. Whereas you could see in control plate the development of compact mass of mycelium, and then development of fruiting body of sclerotia, so in control plate. But in test plates, you could see the destruction of mycelium and the disintegration of mycelium over here. So through which we could count the microorganism, our selected microorganism, they are producing certain substances through which they could able to control this Rhizoctonia solanae. It may be antibiosis, it may be a mycoparasitism, it may be a competition. Antibiosis means the production of antibiotics through which the microorganism to control the pathogenic microorganism. Parasitic means the microorganisms able to produce various lytic enzyme. For example, chitinase, cellulase, amylase, protease, and etc., through which they are able to control the various pathogenic microorganisms. Competition means the microorganism, how fast they could take the nutrient from the petri plates, from the habitat nature, through which they could control the pathogenic microorganism. You could see, and whereas you could see here, see disintegration of mycelium and then compact mass of mycelium. This is the view of our morphology of our selected microorganisms, Streptomyces VS-MGT1014. 1014 in under the view of scanning electron microscope. Yeah, we could see so many colors, the different medium, and then we have done the optimization. Suppose we want to develop a product against the pathogen, we have to optimize our selected microorganism. Our selected both using different factors. See, for example, we are using different carbon sources, different nitrogen sources, different days, different pH. The best growth we find out ISB2 medium is best growth, and then we find out ammonium acetate, it's a best nitrogen source, then sucrose is a best carbon source, and then 6th day is a best growth medium, growth of our in ISB2 medium of our selected isolate via Streptomyces vs MGT 1014. And then we extract the optimized condition, we extract the cell-free filtrate using ethyl acetate. And then the crude metabolite different concentration we check out towards the different fungal pathogen by well diffusion method. You could see the different level of zone of incubation with the different concentration compared to control. You could see a crude metabolite of metabolite which is extracted from Streptomyces. We have selected a microorganism. We identified Streptomyces aeruginosus based on the 16S RDNA sequence analysis. And then we check out to the against sheath blight pathogen Rhizotonia solanae through detached leaf assay method. See, there are 5 different type of treatment. Among 5 different treatment, the Rhizotonia solanae plus culture, they are effectively control the sheath blight of rice, and then cell-free culture which also effectively control. And compared to crude extract or significantly controlled sheath blight of rice 72.0% compared to carbendism. And then we find out some functional characters of our extract, our crude metabolite using different spectral studies, for example UV spectrum, IR spectrum. So we find out some phenazine like compound, because we could compare with the authentic phenazine. You could see this is authentic phenazine UV, this is crude metabolite UV, this is authentic phenazine IR spectrum, this is crude metabolite phenazine. So almost it is equivalent nature of the functional characters, the equivalent to the already established phenazine. So phenazine, we find out phenazine like compound from our selected isolate Streptomyces aeruginosus vs MGT 1014. And then we have done using different concentration, means 5 μg per ml concentration of crude metabolite. We have significantly controlled the Rhizotonia solanae. That thing we observe under the high content screening system. Here you could see even the nuclear disintegration. Nuclear disintegration, you could see due to the treatment of our crude metabolite 5 mg per ml, that is 265 number of nuclei to observe 16 field, 265. Whereas you could see in carbendism 53 nuclei. Whereas in Rhizotonia alone, there are 1761 nuclei are there. See, so our crude metabolite remarkably eradicate the nuclei of Rhizotonia solanae compared to control. Then coming to the Bacillus. Bacillus is also a soil free living gram-positive antagonistic microorganism. This microorganism also control various pathogenic microorganisms. For example, Bacillus cereus, Bacillus laterospores, they are effectively controls various pathogenic microorganisms. See, just you could see the light microscopic observation, the thickening and bulging of the mycelia compared to control. So you just take a pinch of mycelium on zone of incubation, and then you could see under the light microscope with the staining with the cotton blue, you could see over here the thickening and the disintegration of mycelia. Then coming to the trichoderma. Trichoderma is also a very good antagonistic microorganism. It could be used nowadays. People are using very good biofertilizer, very good antagonistic microorganism. It is a very good biocontrol potential organism because it is a very good teutromycete group of microorganisms. It is asexual spore producing microorganism. The soil inhabiting the microorganism. You could see it's a very effective biological control, free living, highly proliferating microorganism, non-pollutive, easily accessible, non-phytotoxic, systemic, and readily biodegradable, cost effective. Moreover, they are also produce various they are also enhance the plant growth by the production of various plant growth promoting substances like auxin, gibberellin, cytokinin, etc. And through which they are produce various defense mechanism, defense enzyme. For example, phenylalanine ammonylase, phenol, and then phytolaxins, through which they could reduce the pathogenic population. Yeah, you could see the dual plate assay of trichoderma against various pathogen. You could see the microorganism that this level is pathogen. So these are trichoderma. This is the pathogenic microorganism. So even this, how they could control, because these microorganisms, they produce various substances. That substances diffuse inside the agar, through which they could control the growth of microorganism. You could, the trichoderma, they could control the various pathogenic microorganism, plant growth promoting substances, and systemic acquired resistance as well as induced systemic resistance. So there are two different methods are there in defense mechanism of plant. But one is systemic acquired resistance. The systemic acquired resistance means it should be released various ethanol, salicylic acid through which to arrest the pathogen. Whereas induced systemic means the microorganisms, the trichoderma, it is enhanced the production of various defense enzyme like phenylalanine ammonylase, tyrosine ammonylase, peroxidase, polyphenol oxidase, and total phenol content, through which they arrest the development of the pathogenic microorganism inside the plant host. There are microorganisms. For example, you may take the Trichoderma viridi. There are so many species of Trichoderma genus are there. Trichoderma viridi, Trichoderma hasianum, Trichoderma coningiopsis. Likewise, there are so many organisms, but it is a highly established antagonistic microorganism. They have a broad spectral activity towards various pathogenic microorganisms. See, the Trichoderma coningiensis, they could easily remarkably control the Potides genera on tomato plants. Yeah, Trichoderma microorganisms, they produce various substances, they produce enzyme, they produce various antibiotics, they can produce so many mycoparasitism, and then systemic resistant, competition, growth promotion, and effective antagonist, and rapid substrate colonization, through which potentially they could act as a biocontrol agent towards various pathogenic microorganisms. Then coming to the fluorescent Pseudomonas. See, fluorescent Pseudomonas is a ubiquitous soil microorganism. In recent years, fluorescent Pseudomonas drawn much attention worldwide because of their plant growth promotion ability and the production of various antibiotics like phenazine, 2,4-diacetyl-fluorochlorinol, pyrrolentrin, and biolutrine. Because the the antibiotics, the fluorescent pseudomonas, they could easily degradable. They could not persist in our environment. Whereas you may use the fungicide, any kind of the chemical, they could persist in our environment and it also cause the environmental pollution. Whereas instead of using chemical pesticide, chemical fungicide, we could use the metabolites from microbial origin. It's a wise choice to control various diseases. So fluorescent pseudomonas, they could be able to produce plant growth promoting substances, various antibiotics, it could be able to produce hydrophore, hydrogen cyanides, through which we could control various plant pathogens. See, this is just for identification. We have done some various genetic diversity of fluorescent pseudomonas from various districts, phylogenetic tree. Then, see how the microorganism they distribute among in between various districts. See, for example, a single group of microorganisms, their population is different from one place to another place. This distribution diversity we could find out through the post-PCR analysis. Genetic diversity we could find out using a method that is post-PCR. Then we could detect the different encoding gene, antibiotic encoding gene. See, each and every gene, they have the specific primer. Using the specific primer, we could isolate the DNA from our antagonistic microorganism, and then our antagonistic microorganism DNA mixed with the specific primer, and then we will do the polymerase chain reaction. After polymerase reaction, we could detect the presence of our interesting gene. See, you could see our isolates of fluorescent pseudomonas, they could obtain the hydrogen cyanide gene. We could find out through polymerase chain reaction with the 586 base pair. Then polycatate synthase 2 encoding gene we could detect from our isolates with 554 base pair using specific primer. And then we detect the presence of adenosine 1-carboxamide encoding gene from our different isolates of fluorescent pseudomonas in different district level. We find out, you could see a single band is present with 500 base pair. Yeah, so the pseudomonas have various species. Each and every species, they could be able to control various diseases. For example, you may take the pseudomonas aureophasian, it could be able to control tallus spot of turf. Pseudomonas fluorescent is able to control the timing of bean caused by Rhizotonia solani. Pseudomonas originose also able to control various diseases, sheath blight of rice caused by Rhizotonia solani. Pseudomonas putida is able to control the fire blight of beer caused by Erwinia amylovora. Then I already told you the biological control or various mechanisms are there: nutrient competition, site competition, antibiosis, and cross communication. Cross communication, I want to mention over here, the microorganism, they could communicate each other through pass on one microorganism to another microorganism through the exchange of chemical signal molecule, through which they could communicate from one microorganism to another microorganism. See, the microorganisms, they able to control. For example, some of the field, maybe, see, could see this is kind of the dual plate assay, but amended with ferrous or ferric ion. Because, some of the field, the iron may be less. But the compensates our microorganism, our antagonistic microorganisms segregate the ion to compensate the ion level in a particular field. through the production of siderophore. You could see the medium itself, the production of senescence we could find out. If a medium, it would produce this kind of the color, it indicates the production of senescence our selected microorganism. You could see the medium itself, the senescence, it will look like a reddish color. Definitely the microorganism able to produce the senescence metabolite. Yeah, various functional characters are there. Various functional characters are produced by the microorganism. For example, phosphate solubilization, siderophore, hydrogen cyanide, indoleacetic acid, protease, pectinase, chitinase, cellulase, and amylase. So these are all the functional genomics. We could find out those kind of these respective genes are present or not. If it is present, definitely they are produced. For example, a Chi gene is present in a particular microorganism. They are able to produce chitinase enzyme. So the chitinase enzyme is a very important hydrolytic enzyme. They are able to control various pathogenic microorganism. Then coming to the biofilm, yeah, microorganism, they are developed through the segregation of hexapolysaccharide. They could develop a compact plastic layer in and around roots of plant roots, through which the microorganism, they develop a layer, a plastic layer, they are called biofilm. So we could detect from our microorganism the production of biofilm through the estimation of hexapolysaccharide. You could estimate the hexapolysaccharide estimation through microtitrate plate assay. We are used to develop different kinds of our antagonistic microorganism. Among different microorganisms, we find out BSNGT18, they produce more amount of hexapolysaccharide compared to other microorganisms. Yeah, we are done the different optimization. We use the different carbon sources for biofilm formation. Among different carbons, the fructose, they produce more biofilm. It means it produce more hexapolysaccharides, through which they could develop more biofilm layer. Then the yeast extract is a best nitrogen source for biofilm formation by BSNGT8. So you could see the biofilm development, biofilm layer development under the high content screening by our microorganism BSNGT18, carbon and nitrogen sources. You know, the A is a control, then B carbon source, C nitrogen sources. You know, when using this carbon and nitrogen sources, then compact with both carbon and nitrogen sources, here intensity of biofilm formation you could see over here compared to control. Then different pH also play a physical factor, is also very important. pH 7 is a best physical factor for establishment of biofilm formation by our microorganism BSMG28. Then through the motility assay also we could find out gram staining, flagella motility and twitching motility, through which, because you know, through the twitching motility there is a specific medium of there, and that specific motility itself you could find out the formation, spreading of the colony, through which we could find out then flagella motility. If it is through how the microorganism they could move from one place to another place due to the presence of flagella. So they could move from one place to another place because they could find out they need, there is some particular need, particular nutrients or place, one particular place, they could move from one place to another place because of the presence of flagella. Then exopolic estimation. Then we also done the under the scanning electron microscope using our plants. For example, we are growing inoculation with our selected microorganism BSMGT8 on green gram. Then we check out the section of, you have taken the section of green gram roots. We could see under the scanning electron microscope, you could see the formation, compact and development of the biofilm layer of microorganism. Because of a selected microorganism, it is also act as the endophytic nature. You could see here the microorganism with the development of a layer. That layer is called exopolysaccharide. So through which it is we find out, we could clarify it. Our selected microorganism, that is VSMGT8, they could say very successful and significantly they produce biofilm layer by the production of exopolysaccharide. And then black gram roots, you could see over here the colonization of microbe with compact development of layer with the production of exopolysaccharide. Then coming to the rice seed. Our other kind of research we have done, that is how to control the seed blight. As I already told you, seed blight disease of rice caused by Rhizoctonia solai, it is a second most devastating disease. It could cause yield loss about 2 to 69% in India. So cultural control, it is not effective. Breeding for resistance not successful because of the unavailability of seed blight resistant genes. You could see this is a seed blight of rice symptom on rice plant caused by Rhizoctonia solani. So as already told you, initially the Rhizoctonia solani, it is look like white color, then it is turned brown color and then develop various diseases. You know, the recent years, the fluorescent pseudomonas have drawn much attention and they effectively control the plant disease by the production of siderophore, antibiotics, phenazine, 2,4-diacetylfluoroquinolone, pyrrolitrin, biolutrin, volatile compounds, hydrogen cyanide, lytic enzymes, and they also significantly promote the plant growth by producing phytohormones. You know, because the fluorescent pseudomonas are drawn much attention worldwide because of their plant growth promotion efficiency and biocontrol potential. Fluorescent pseudomonas, they could, moreover, in our Indian soils are rich in microbial diversity and wealth of indigenous microflora, but has not been fully investigated. Hence, there is an immense scope for identification of novel metabolites and novel microorganisms. So based on that, we have chosen the following objectives: isolation, identification of effective fluorescent pseudomonas against Rhizoctonia solani from rice rhizosphere, determination of growth promotion activity, evaluation of top coder formulations against seed blight under greenhouse condition, determine the mechanism of biological control, then purification, characterization of antifungal metabolite, then evaluation of purified metabolite against seed blight under greenhouse condition. See, one of my student collection of soil sample from rice field. See, the collection of the rhizosphere soil, we isolate the fluorescent pseudomonas by serialization technique on Kingsby agar medium. You could see over here, your fluorescent colonies are present on Kingsby agar medium because the selection of isolation of fluorescent pseudomonas, there is a specific medium is present, that is Kingsby agar. You could see this is a purified colonies of fluorescent pseudomonas on Kingsby agar medium. All the organism, all the isolates we check out towards our rocket pathogen Rhizoctonia solani. You could see the various level of zone of incubation. So totally we isolate 70 soil sample from different districts of Tamil Nadu, starting from Kanyakumari to Theni. Each and every district we have collected 10 rice rhizosphere sample. So totally we isolate 671 fluorescent pseudomonas. From 67 fluorescent pseudomonas, we find out 87 fluorescent pseudomonas showed antagonistic activity against Rhizoctonia solani with the different level of zone of incubation from starting from 0.2 to 2.8 centimeter. See, among 671, we find out a potential organism, that is only one microorganism, that is we designate number, that is MML2212. You could see here the fluorescent colonies of selected isolates on Kingsby agar. We also test towards the various microorganisms, various pathogenic microorganisms, because the fluorescent pseudomonas they have a broad spectral activity against various microorganisms like alternata, Pyricloria oryzae, Fusarium oxysporum, Fusarium budum, Macrophomina phaseolina, and Carularia lunata. So these are the zone of incubation from 14 millimeter to 28 millimeter compared to control. See, this is also, we have done our selected isolates. Our selected isolate MML2212, they effectively control various pathogens by their cell-free culture filtrate by bulk diffusion method. So you could see from the zone of inhibition, we have taken a single piece of mycelium, and then we strain with the cotton blue, and then we have see under the light microscope. You could see over here the disintegration of mycelium because of our cell-free culture filtrate of our selected isolate MML2212. And then you could see the mycelial inhibition zone, the abnormal growth, and the mycelial tips appears to be cheesed. You could see appears to be cheesed, and then there is no sign the development of the sclerotium. Whereas you could see in control plate, a compact mass of mycelia is developed and indicate the development of sclerotial development. You could see over here. And then we also tested out the cell-free culture filtrate against Rhizotonia solanae on their sclerotia and their fruiting body. And then we have using our cell-free culture filtrate, and then we introduce the uniform size of fruiting body of sclerotia, and then we give the incubation time from 4 to 24 hours. You could see here the germination of sclerotium from 4 hours only 40%, whereas in 8 hours 20%. From 12 hours onwards, there is no germination. 100% of germination is inhibited compared to control. Then we identify using physical and chemical various 16 ordinary sequence analysis and physical characterization. We identify a molecule organism Pseudomonas aeruginosa MML2212. Then we identify Pseudomonas aeruginosa MML2212. And we also done some functional plant growth, self-filtrate, as well as the culture of Pseudomonas aeruginosa MML2212. They significantly enhance the plant growth of rice, as well as they significantly enhance both the sterile and the non-sterile soil, and is also significantly, hello? So we optimize the production of indole acetic acid. Indole acetic acid is a plant growth promoting substance. Our selected isolates MML2212, they significantly produce the indole acetic acid using Kingsby broth, and then we find out detect the presence of the indole acetic acid using thin layer chromatography. See the thin indole acetic acids with 0.78 from our selected isolate. It is coincide with the top authentic IAA. As I already told you, so microorganisms, they are compact, they are consider the level of iron in our agriculture feed by the production of siderophore, and some of the microorganisms, they able to produce the hydrogen cyanide through which they control various pathogens. Where you could see the you have to take you take Botman number filter. Hello. Hello. So our microorganisms, they able to produce the hydrogen. As you can see, the yellow color of Botman number paper, it is changed to reddish brown. It indicates the. Then finally we go for the purification and characterization of metabolite from our selected isolate Pseudomonas aeruginosa MML2212. We grow our microorganism in fermenter conditions 2 liter. After 36 hours we harvested and then we centrifuge it at 10,000 RPM. Then we extracted the supernatant using ethyl acetate extract. Then we are using the column chromatography. We purified a metabolite. For a metabolite, there are 3 fractions. Among 3 fractions, we found out a yellow needle from fraction 2. That yellow needle compound we crystallize. Then finally, we find out a compound molecule that is finishing. So you could see the purification of metabolite produced by Pseudomonas aeruginosa through column silica gel column chromatography. We find out initially, as I already told you, they find out the 3 compounds, that is 0.69, 0.63, 0.46 the compound. Then we again go for purification using column chromatography using 230 to 400 best column. From that we find out a single colony, single purified colony with the RF value 0.54. Then we have done various spectral studies using IR spectrum and mass spectrum. See, through mass spectrum we find out mass values, and then we find out through NMR, H1 NMR and C13 NMR, we find out various delta values, and then we find out various singlet and delta values from both HNR and the C13 NMR, and then we crystallize our purified compound using our various solvent system. For example, we have done 3 different solvent system. For example, acetone, chloroform, and ethyl acetate. But we got a very good fine crystal using chloroform. So using chloroform, we find out this yellow needle crystal. Then we have go for the XRD study. Based on that XRD study, alpha, beta, gamma values, we find out the crystal structure of our purified molecule, that is phenosine 1-carboxamide. The IUPAC name 510 dihydrophenosine 1-carboxylic acid amide, the formula C13 H9N3O. But this is the first report on crystal structure of phenosine 1-carboxamide. So this is a packing structure of phenosine 1-carboxamide. This is the hydrogen bonding pattern of phenosine 1-carboxamide under the X-ray diffraction meter. So this is a first crystal structure. So then we plan to deposit to Cambridge Crystallographic Data Center. They allocated a number, that is CCDC617344. Then okay, so once we purified a molecule, that molecule, how it could be active, how it could be useful towards our target pathogen. See, target pathogens, then we go for the minimal, identify the MIC value, minimal inhibition concentration. From 5 μg per ml concentration onwards, it is significantly control the pathogenic microorganism, and then it is from 12 hours of soaking of the fruiting body of sclerotia, it is significantly controlled. 100% inhibit the sclerotial germination, and then through the poison plate technique, we find out 5 μg per ml of phenosine 1-carboxamide, it is effectively control the pathogenic microorganism Rhizotonia solani compared to carbapenem control. And then it is also control various microorganisms using 5 mg per ml of concentration of phenazine 1 carboxamide and compare with the carbendazim with similar concentration by well diffusion technique. Then our purified compound phenazine 1 carboxamide, it is significantly control the another bacterial rise pathogen, that is Xanthomonas oryzae. Using different concentration, it is effectively controlled from, it start from 5 mg per ml of concentration compared to reformycin. And we have done some randomized block design greenhouse experiment using our selected microorganism Pseudomonas aeruginosa MML2212 and phenazine 1 carboxamide, minimal inhibition concentration 5 mg per ml and similar concentration of carbendazim. Our purified metabolites and various treatment we have done using seed treatment, root tipping, soil application and foliar spray of our isolate Pseudomonas aeruginosa. They significantly enhance the plant height, seed blight lesion, length, and it is suppress the disease incidence compared to our finishing. But even though our purified metabolite phenazine 1 carboxamide, it is almost equivalent to seed treatment, root tipping, soil application, and foliar spray. You could see over here the plant height is 72.3 and seed blight lesion length 12.6 and disease incidence 17.4 compared to carbendazim. And our purified metabolite as well as our selected microorganisms Pseudomonas aeruginosa MML2212, they are remarkably increase the number of tillers, number of productive tillers, and 1000 grain weight and grain yield. So you could see over here the seed treatment, root tipping, and soil application and foliar spray is 20, number of tillers, number of productive tillers 18, 1000 grain 28.5, grain yield 38.7. Whereas in our purified metabolite phenazine 1 carboxamide at few mg per ml concentration, they are highly enhanced the number of tillers, number of productive tillers, 1000 grain weight, and grain yield compared to carbendazim. Coming to the summary, totally we isolate, there are 671 different isolates of fluorescent Pseudomonas, of which 87 show the inhibitory activity against Rhizoctonia solanae with a different level of zone of inhibition from 0.2 centimeter to 0.8 centimeter. Our isolate designated as MML2212, they are exhibited the maximum inhibitory activity against the Rhizoctonia solanae and other phytopathogen. The isolate MML2212 was identified Pseudomonas aeruginosa based on the morphology, biochemical, and 6-ness ordinary sequence analysis. Pseudomonas aeruginosa, they are significantly increase the seed germination and other growth parameters. The application of seed treatment, root tipping, soil application, and foliar spray, they are significantly reduce the seed blight incidence and enhance the yield parameter in rice under greenhouse and field condition compared to foliar spray of carbendazim and control. Our isolates, Pseudomonas aeruginosa, MML2212, produce redophore and hydrogen cyanide. A compound was purified from culture filtrate of Pseudomonas aeruginosa and that was crystallized. It is odorless yellow crystal with a molecular mass 223.3 and its melting point between 220 degrees C to 222 degrees C. The purified compound is 5,10-dihydrophenazine 1 carboxylic acid amide with an empirical formula C13H9N3O. The minimum inhibitory concentration of our purified metal purified compound is at 5 mg per ml against Rhizoctonia solanae. However, I didn't show the dark formulation. Through the dark formulation, our purified organism Pseudomonas aeruginosa MML2212, they are remarkably controlled seed blight of rice compared to carbendazim and purified metabolite phenazine 1 carboxamide under greenhouse condition. However, foliar spray of phenazine 1 carboxamide was significantly effective than the commercial fungicide carbendazim in controlling seed blight disease of rice. Thank you. Hello. Hello. Hello.
Good afternoon. Good afternoon, sir.
Good afternoon.
Sir, your presentation was really nice.
Thank
and
you,
we
madam.
could gain knowledge on Rhizoctonia solanae and other biological control agents.
Okay,
I am
madam.
from the botany field, so the presentation was really very interesting and excellent. Thank you, sir.
Thank you, madam. Sure. Anybody have any doubts, sir? Hello. Hello.
Sir, good afternoon, sir.
Good afternoon.
Sir, your presentation was excellent. Though I am not a botanist, I could understand a lot of things out of that. I am a physicist basically,
Oh.
but trying to work some bio applications actually. But what I understand with your presentation is maybe the bacteriums or the species what you take. Is it necessary to sense in advance? That is, what is it possible to identify these microbes only after they affect the plants, or can it be prevented before multiplication or something like that? Is there any methods for that?
No, sir. Before affected, we could identify the microorganism up to the species level, madam.
Pardon? I don't get you clearly.
So, see, for example, suppose we want to identify an antagonistic microorganism.
Yeah.
That antagonistic microorganism we could identify up to the species level using 16S RNA sequence.
Okay.
So once we find out a novel antagonistic microorganism, those microorganisms we could use to control various pathogenic microorganisms under both the greenhouse as well as the field condition.
Okay. But it is only through the DNA sequencing we can do it.
Yes. So
there is no
that is nowadays advanced methods already came. Using 16S RNA, we could easily find out. No need to go for our traditional technique, biochemical assay.
Okay. I have another interest, being a physicist.
Yes.
Taking up any, see, in a biomaterial, this bioimpedance analysis is coming up rather fast in a biological, like human or anything. Is there any such employment of these bioimpedance techniques in botany or identification of such species?
Yes, so many laboratories nowadays established for identification of microorganism.
Analysis?
By microorganism analysis, and then so many biofertilizer industries come up using my antagonistic microorganism. Yeah, through which people are getting so many recruitment.
Okay.
Yeah.
Thank you, sir. Thank you so much.
Thank you.
Related to Shanmugaiah's session, you can ask.
Hello, sir.
Hello. Yes.
Am I audible, sir? Sir, I want
Yes.
to ask. Yes, sir. Sir, my name is Riddhi, and I'm basically a microbiologist.
Okay.
And I want to ask that if plant is infected by virus, then what will be the procedure for identifying viruses? Just like for micro, we do 16S RNA sequencing. So
See,
for
any
viruses,
kind of.
yes, sir.
Hello.
Yes, sir.
For viruses, any microorganism, for example, not 16 is only for the prokaryotic microorganism. Whereas in eukaryotic microorganism, you can go for 18S.
So what about virus? If because I'm working with one of my colleague, he's a botanist, but I haven't done work in virus identification. So what we were thinking in our result, we got that plant is affected by virus.
Where 16S the only, madam.
16 okay.
Yes.
So sir, where to send the sample and how, what will the sample?
What?
What will be the procedure for sending sample, and is there any
You
university?
isolate the DNA from your respective organism, your interesting organism, and then we do the PCR, polymerase chain reaction. After then the PCR reaction, you may send to the sequence industry.
Sir, do you suggest any good sequencing industry where the identification is like good? Because in past we had experience and that with my guide during PhD, if we send sample to one laboratory and if we compare data with our biochemical test in laboratory, they are completely different. So my guide sent sample to other laboratory, and at that the identification was completely different. So do you suggest any where sequencing is we can send sample and sequencing done?
Then you have to find out another laboratory, and then you may send to even foreign countries also. You may send your samples.
Okay. Thank you.
Sir, this is Tamilnadu.
Hello.
Hello, sir. This is Tamilnadu, sir.
Tell me, madam.
Sir, I belong to computer science, sir.
Okay, madam.
I know what are the research opportunities related to this plant disease detection for our field, sir?
What are the
Research opportunities like plant disease detection.
Using this plant disease reductions, nowadays people are very much interested to cultivate various flowering plants, various cereals plants, various kind of plants. See, but the plants, they are affected by both biotic and abiotic factors. I has already told you, for example, various fungi, bacteria, and viruses. But these COVID viruses is eliminated through the not using chemicals. We have to use alternate use, that is microorganism. So through which the people are developed so many biofertilizer. We could identify a novel microorganism. So many, through which they could develop so many biotechnology industries. Once they, through the development of biotechnology industry, they could identify the novel metabolites. See, for example, you may take the phenazine is one kind of established molecule. That molecule, there are 15 different types of derivatives already identified. But still there is a scope, because even if you take our Indian, because India is a very big diversity. Indian soils are very big diversity of microflora. But even there is a scope for identify a novel metabolite to study the isolation and purification of compound biomolecule from the microorganism.
Sir, what kind of disease is the most widely needed one, sir, for research?
See, in India and China, we are majorly we are taking rice.
Yes, sir.
So rice is affected by various pathogenic microorganisms. For example, best is you may take the Rhizotonia solani. That is a fungus. It belongs to the Tetromyces group of fungi. This Tetromyces group of fungi they will be able to survive even a decades in our soil. Once it is entered in our agriculture field, it could not easily degradable. You could not eliminate. You could not destruct that pathogen. For the destruction or elimination of this pathogen, it will go for up to a decade. So it is a highly economically important pathogen. So for the control of this pathogen, we could identify. Those days people are using. There is a systemic fungicide that is carbendazim and mango sap for the control of Rhizotonia solani. But to keep on applying this kind of fungicide, they cause the environmental pollution. And while they are also not easily degradable, they persist in our environment, in our agriculture field. So for the eradication, we need to alternate use. You can use the biocontrol organism, microorganism. The microorganisms not persist in our environment. They could easily degradable. Got you, madam?
Yes, sir. Thank you, sir. Thank you so much, sir.
Is there any other question from the participant side?
If there is no questions,
yeah,
I extend my heartfelt thanks to Dr. Shanmugaiah. Thank you, sir. Thank you so much for your excellent presentation today, sir.
Thank you. I also want to say 2 words. I am very much thankful to organizing committee members to give a wonderful opportunity to present our research findings in front of this wonderful audience. So I very much thankful to all organizing committee members. Thank you, one and all.
Thank you, sir. Thank you so much, sir.
Thank you.
If there is no doubts, we'll wind up the session and we will meet tomorrow for day 12 session. Thank you.
Thank you.